Hello everyone, welcome to Mediverse Atlas. In today's video, we are going to talk about the neuromuscular blockers. So, to make your concepts clear, do watch the video till the end. So, neuromuscular blockers. What do we understand by the name neuromuscular blockers? So, as the name suggests, there are some blockers which block the transmission of nerve impulse leading to the impaired contraction of muscle. Okay, so these are nothing but the neuromuscular blockers. Now, this transmission of nerve impulse can be blocked by two ways, either by presynaptically or either by postsynaptically. So, neuromuscular blockers can be presynaptic blockers as well as postsynaptic blockers. Now, what are these presynaptic blockers? So, there are two presynaptic blockers. Those are botulinum toxin and hemicolinium. Whereas postsynaptic blockers are classified as competitive blockers and depolarizing blockers. Now, competitive blockers are curare and galamine, and depolarizing blockers are reversible acetylcholine esterase inhibitors and irreversible acetylcholine esterase inhibitors. Okay, so this is all about a brief intro which we are going to talk about in this video. Now, first we'll be discussing about the presynaptic blockade. Now, what do we exactly understand by this presynaptic blockade? So these blockers, they block the presynaptic events leading to the impaired calcium influx, which causes decrease in the release of acetylcholine. Now, when we look at the neuromuscular junction, if this is a presynaptic membrane, if this is the postsynaptic membrane, we have some calcium channels which are present. So these calcium channels causes the influx of calcium and leading to the release of acetylcholine into the synaptic cleft. So these presynaptic blockers, they block the transmission of these or the influx of these calcium, leading to the decreased acetylcholine. So this is the basic point here. Now, in this presynaptic blockers, first we'll be talking about the botulinum toxin. Now, this toxin, it is released by a bacteria which is known as Clostridium botulinum. Okay, this Clostridium botulinum, it releases the botulinum toxin leading to the presynaptic blockade. So this toxin causes a disease called as the botulism. Okay, now this is very dangerous. Okay, this toxin is very dangerous. The lethal dose for adult human is just 2 to 3 microgram. So this is the lethal dose for a human. Now, what exactly these botulinum toxins do? So this botulinum toxin, they cleaves the snare proteins which are required for the fusion of vesicles. So there are some proteins called as snare proteins. These snare proteins help in the fusion of vesicles, okay, to the presynaptic membrane. So the vesicles, they fuse to presynaptic membrane and they release acetylcholine, right? So this botulinum toxin, they cleave the snare proteins. If there are no snare proteins, there will be no fusion of vesicles. So there will be impaired release of acetylcholine. Now what are the snare proteins? There are three snare proteins: SNAP25, synaptobremin, and syntaxin. So these are the snare proteins which are present. Now this botulinum toxin, like they prevent the release of acetylcholine and causes flaccid paralysis. This is very important. MCQ can be asked. So which paralysis is caused by the botulinum toxin? The flaccid paralysis is caused. Now, highly diluted doses are used for therapeutic and cosmetic purposes, and they are also used in the treatment of achalasia cardia, and they are injected to extraocular muscle to reduce strabismus and blepharospasm. Okay. Now, hemicolinium, the second presynaptic blockade. Hemicolinium, they inhibit the uptake of choline by presynaptic terminal, which results in the decrease of acetylcholine, and end plate potential decreases and muscle contraction do not take place. So what exactly happens is, after the release of the acetylcholine into the synaptic cleft, suppose this is synaptic cleft, after the release of acetylcholine into the synaptic cleft, there is an enzyme called as cholesterase, that is ACHE, acetylcholine esterase. This causes the cleavage of acetylcholine into acetyl and choline. Okay, this choline is again uptaken by the presynaptic membrane for the synthesis of ACH, whereas acetyl is metabolized. Now, this hemicolinium, it blocks the uptake of choline by the presynaptic membrane. So if there is no uptake of choline, there will be no synthesis of acetylcholine. Okay, so this is the action of hemicholinum. Next, the postsynaptic blockade. So the postsynaptic blockade, first we'll be talking about the competitive blockers. So these agents compete with acetylcholine to bind with acetylcholine receptors. So these acetylcholine receptors are present on the postsynaptic membrane. So these competitive blockers, they compete with acetylcholine to bind to these receptors. So the first one is Curer. So this Curer is a plant product. It is used in the hunting of animals and it causes respiratory paralysis, the paralysis of respiratory muscle leading to asphyxiation. Okay, so this Curer competes with acetylcholine to bind with acetylcholine receptors. So this Curer binds to the acetylcholine receptors, leading to the paralysis of respiratory muscle, asphyxiation and death. Next, galamine. This galamine is very important. Okay, so this galamine, it is given before surgery to relax the skeletal muscles. It reduces the dose of anesthetic drugs and decrease the risk of bleeding and other complications. So this is an important neuromuscular blocker used in the medical field. Okay, next, the second category is the depolarizing blockers. So these depolarizing blockers, they inhibit the activity of acetylcholine esterase. Okay, so in depolarizing blockers, first we'll be discussing about the two important depolarizing blockers, that is succinylcholine and carbamylcholine. So these are also depolarizing blockers. So drugs such as succinylcholine and carbamylcholine have same biological activity of acetylcholine. So they are not hydrolyzed by the acetylcholine esterase. Okay, normally acetylcholine is hydrolyzed by acetylcholine esterase, right? These two drugs, they have the same activity of the acetylcholine, but they are not hydrolyzed. That is, acetylcholine esterase cannot act on them and cleave. Okay, so they inhibit the activity of acetylcholine esterase. Now, these drugs bind to the acetylcholine receptors, causes depolarization, and they maintain depolarization and block the subsequent action potential. As discussed here, suppose if acetylcholine is not cleaved, what happens? Depolarization takes place, right? If depolarization takes place, there will be contraction of muscle, and these two drugs, the carbamylcholine and the succinylcholine, these are not, this acetylcholine esterase cannot act on this succinylcholine and carbamylcholine, right? So depolarization is there, still there, it continues. Okay, so the action potential will be generated and there will be contraction of muscle. Okay, and depolarization is maintained. Okay, just remember the succinylcholine or carbamylcholine has the same activity of acetylcholine, and they are not acted by acetylcholine esterase, and we have reversible acetylcholine inhibitors. So these two are the reversible acetylcholine esterase inhibitors. Those are neostigmine and physostigmine. Okay, and the concentration of drug decreases over time and block is removed after some time. That's why these are reversible. So these neostigmine and physostigmine are also used in the treatment of myasthenia gravis. Okay, neostigmine and physostigmine. So these are reversible. Why they are reversible? Because their concentration decreases over time and the drug action is decreased. Next, irreversible acetylcholine esterase inhibitors. So these are organophosphorus compounds, that is pesticides or nerve gases, so that such as DHP, serine, and they bind to acetylcholine esterase tightly and irreversibly. These are very important in second year pharmacology also. You will be studying about these compounds, so make sure that your basics are clear. So these organophosphorus compounds are nothing but the poisoning agents, like many used for suicide, no? So these pesticides and nerve gases, and these bind to the acetylcholine esterase tightly. Okay, so if the acetylcholine esterase is bind tightly with these compounds, there will be decreased action of it, right? So there will be lack of hydrolysis of acetylcholine, causes sustained depolarization and blocking further action potential. Okay, like muscle contraction do not takes place here because of the sustained depolarization and blocking of further action potential. Poisoning with these agents leads to skeletal muscle paralysis and death from asphyxiation. And immediately, if detected early, atropine can be given as antidote. So this is all about neuromuscular blockers. Hope you enjoy the video. If you have any doubts, please do comment in the comment section. Do follow our Instagram and Telegram channels, and please do subscribe, like, share. Thank you.